अरे यार क्या हुआ, क्यों रुक गया तू?
ये देख दीप, जल्दी आ, मैंने क्या ढूंढा।
अरे वाह, क्या चीज ढूंढी है तूने तो।
अरे ये है क्या?
क्या है?
अरे यार क्लास में सो रहा था क्या, ये कोलोकेसिया का डिसीज है जो फोलियर पैथोजन है, वो पीटियोल और कॉम को भी अफेक्ट करता है।
अरे मुझे लगता है कि ये तो वो फाइटोप्थोरा लीफ ब्लाइट है ना, जो सर ने हमको पढ़ा था क्लास में, उसके सिम्टम्स लगते हैं।
हाँ हाँ, ये देख, वैसे ही दिख रहे हैं। ये देखो, लोअर सरफेस पे वाटर सर्कुलेशन भी है।
हाँ है ना।
ये क्या क्या ये वाटर सर्कुलेशन है।
और स्पॉट में कॉन्सेंट्रेट रिंग जैसा भी कुछ दिख रहा है।
दिखाओ।
ये वाला
और पत्ते भी देखो फ्लैग की तरह झूल रहे हैं।
हाँ मिल जाएगा।
और ये देखो, स्पॉट हॉल वाले सिम्टम्स भी दिख रहे हैं।
अरे यार,
ये देखो, स्पॉट की साइज आपस में मिल रही है।
दिखाओ।
ये देखो, लीफ की अपर सरफेस की स्पॉट के चारों ओर येलो येलो भी दिख रहा है।
और ये देखो, पोट की शुरुआत लीफ मार्जिन से हो रही है। और यहाँ एक व्हाइटिश कलर का रिंग दिख रहा है, ये स्पोरेंजिया का लग रहा है।
हाँ है ना मुझे।
ये देखो,
तो ध्यान से देखो, इन्फेक्शन साइट में से रेडिश ब्राउन कलर का कुछ लिक्विड जैसा निकल रहा है।
इतना सारा प्रूफ देखने के बाद मुझे अब लग रहा है कि ये फाइटोप्थोरा का ही सिम्टम है।
इसे देख के तो हम फाइनल जजमेंट नहीं दे सकते।
तो हालो, હવે હું તમને બધાને લેબમાં લઈને સાબિત કરાવું।
Now we are going to prepare a slide. First we take a slide, then put a drop of water on it. Then we will transfer the spore with brush. Then it cover with carefully cover slip.
slide is prepared. We will check it under microscope.
Flashback to 1943. Brown spot of paddy caused a serious famine in Bengal with death of over 2 million people. Going back further, coffee rust by Hemilia vastatrix ruined the whole economy of Sri Lanka during 1867 to 1899. These events emphasize the impact of plant diseases and why it becomes important to study them in vivid detail. Hello and welcome. This is Sejal Singh, and along with my fellow classmates, we are going to present an informative video on plant pathology under the course title Diseases of Field and Horticultural Crops and Their Management. In this video, the disease illustrated is Phytophthora leaf blight of colocasia. We have tried to depict it in a clear, simple and fun way. So let's jump into the video.
Taro leaf blight is highly infectious plant disease. It is characterized by formation of large brown lesions on leaf. Oomycetes leach nutrient out of the leaf via hysteria and create a white powdery rings of sporangia. These pathogens grow best in high humidity and high rainfall environment. Taro leaf blight epidemic of Samoa in 1993 is good example of its heavy destruction by its pathogen.
Myself Abhishek Ajudian. Now we discuss about the life cycle of Phytophthora leaf blight. First one is germination. Sporangia require free water in which to germinate. Can germinate in either of two ways depending upon the temperature. Under the warm condition 28 to 30 degrees Celsius, sporangia germinated directly by the germ tube that can infect the leaf. This is a slower process than juice for production can take 5 to 6 hours for sporangia to germinated. The proportion of a sporangia germinating directly is generally much lower than for those forming juice spore.
Myself, Thakur Vivek. Now I am explaining about the infection. Infection occurs both sides of the leaf surface, upper leaf surface and the lower leaf surface. In the infection, first, germoplasma penetrate in the epidermis layer of the leaf through stomata and spread through the inter and intracellular leaf tissues. Most of the infections occur during the midnight and the morning time when the dew is present. At the daytime, when moisture content is high in the environment, then infection occurs.
Myself Gaurav Wadher, I explain symptom development. Initial symptom appear within 36 hours of infection. A small water soaked in the leaf surface. The fungus is normally most active during the night and lesion morning. Distinctive soak margin weight mass of sporangia. During cooler and rainy, lesion can continue expand during the day. The growth of the fungus within the tissue of plant strongly affected by temperature. To optimum temperature for the growth of the fungus, in vitro approximately 25 degrees Celsius. At the 35 degrees Celsius, symptom is stopped. During dry and cool weather, common lesion of the field is expanding. The most rapid expansion lesion when cool and showery weather allows fungal growth tissue both night and day.
Myself, Pokya Chintan. Now I explain the sporulation. Sporulation normally occurs only in the zone of active fungal growth at the margin of the lesion. Sporodiospore emerges either through the stomata or directly through the epidermis on both surfaces of the leaf. Under optimum condition, sporulation can take place at the margin of a lesion in less than 3 hours. Sporulation normally peaks between midnight and dawn with no sporangia produced during the day.
Myself, Deep Sakwada, I explain dispersal and spread. Sporangia and juice spore in rain splash and wind-blown spray are the principal source of spread within and between plant. Rivulet of dew and exudate droplet carrying and juice spore are in fact important mechanism of spread on and between leaves of the same plant. Sporangia are not released into the air on drying. Long distance dispersal of the organism occurs only by movement of infected plant material, leaves, petiole, infected combs, etc.
Hello everyone, my name is Timbadia Dishant and now I discuss about the survival of the pathogen. Primary mode of survival is continued recycling of the pathogen. It is not well adapted to long-term survival in the absence of the living host material. Fungus cannot survive on hypa in the soil. Under material circumstances, large number of sporangia are washed in the soil. Most of these discharge juice spore.
Hi, myself Suthar Santi. Now we are going to discuss the management practices regarding to this disease. Remove the infected leaves and comb. Avoid the planting in shading locality and proper drainage must be ensured. Use the resistance variety like Ahina, Pusha and Shakin 5. Healthy comb should be selected for the planting. Crop rotation which reduce the disease intensity.
In the early stage, spotted leaves should be removed and destroyed. Field sanitation may decrease the inoculum level early in the season. Spraying the Bordeaux mixture and also spraying the metalaxyl at 15 days in 3 weeks intervals.
આ વિડીયોમાં આપણે જોયું તે પ્રમાણે આ એક ફૂગ રોગ છે જેનું નામ ફાઇટોફ્લોરા બ્લાઇટ છે જે ફાઇટોફ્લોરા કોલોકેસિયા દ્વારા થાય છે. આ રોગ જમીનમાં રહેલા વધુ ભેજ, વરસાદી વાતાવરણ અથવા ભેજવાળું વાતાવરણ અને ગરમ તાપમાનના કારણે વધે છે. અનુકૂળ વાતાવરણમાં આ રોગથી સંપૂર્ણ છોડ મરી જાય છે, જ્યારે અનુકૂળ વાતાવરણ ન હોય ત્યારે 25 થી 35% નુકસાન કરે છે. રોગના મુખ્ય લક્ષણોમાં કથ્થાઈ ટપકાઓ, દેખાવો ઉપરના ભાગે અને વોટર સોપ લેસરનો દેખાવ પાંદડા નીચેના ભાગે જોવા મળે છે. આ ડાઘ ધીમે ધીમે ભેગા થાય છે અને પાંદડાઓ મરી જાય છે, જે જાળા જેવા દેખાય છે. નિયંત્રણ: રોગવાળા પાંદડાઓ કાઢી નાખવા, ખેતરને ચોખ્ખું રાખવું, પાકની ફેરબદલી કરવો, પ્રતિકારક જાત જેવી કે પૂનમ, રીના, પૂષાનો ઉપયોગ કરવો, બોડેક્સ મિક્સરનો છંટકાવ કરવો.